Okay.
We are now live.
We're still waiting.
Hi, from Palo Alto.
We're still
We're
waiting.
still waiting? Have you seen this?
I'm up on the stream side.
Why don't we wait until then it says we're live, because I don't believe we're live.
We can't check verified voice until.
Okay.
it is
That's
out.
true. Yeah, let's do a sound.
All right. A sound check. Okay. Yeah. 1, 2, 3, 4.
Live.
Okay,
Testing,
now we're live.
testing. Okay. All right.
I hear chuckling.
All right. Okay. Hello, everybody.
Now comes the question. We were alive when you asked the question, Brad. I just got.
All right. Okay, let's get started. This is CH Ting, and this is Silicon Valley Fourth Interest Group in our morning session. And the topic today, just like last month, it's code in long non-coding RNA. It's just a continuation of the work that I've been doing for more than a year now. Okay, let's just get started. This morning, I have just two topics. The first one is the Python is fourth in C, okay? So, and that's my excuse of switching from fourth to C in all the analysis. And then I will talk about the problems that I have in the long non-coding RNA analysis. Python is fourth in C. And the Python interpreter, or what they call idle, feels just like fourth. It's an interpreter. You type things, it does the work. And in fourth, we put everything on a stack. So you often you look at stack and to see what's going on. And the Python puts a result just on top of stack. So if you do anything, it will print out what the result or the most likely the some status or some result. If you do 1 + 3, it would display 4, just like fourth. But the Python kind of use the syntax very much like C. So I guess for a fourth user, we just have to switch the postfix kind of the thinking into infix thinking, and you're right at home. And the most important thing is that the Python lists. It's Python list is almost a basic data structure used in Python. So everything, generally it will convert everything into a list. And then you can just handle everything from the list and do other things. So the list is extremely powerful, and it can handle multidimensional data. And you can mix numbers with strings and any other kind of stuff. So the the list is extremely powerful. And I found I don't have to go off to other data structures to do my genome analysis. So everything, I think almost everything's done in lists. At one point, I thought Python has a very nice structure of dictionary. Okay, and I was thinking, oh, sometimes I could use a dictionary for some analysis, but it turns out that dictionary is much more restricted than lists. So the amount of data you can put into a dictionary is much less than what you can do with lists. So finally, I gave up on dictionary and just concentrate mostly on lists. And the file system is also extremely flexible. You can open up lots of files. You can write things out, and you can read things back and do all kinds of things. So the file system is very important in the if you have a complicated analysis. Sometimes you want to you don't want to have everything in memory. So part of it you can write it out. And sometimes it's also advantageous because when you do analysis, some of the data you want to keep, some of the data you want to throw away. And it's kind of awkward to do it in memory. So why not just write out all the data you want to save out to a file, and later on in the next step you can read them back. So I'm very happy with Python. So the originally many of the critical routines were written in Forth, and all these were converted into Python, and it's not difficult. And it is very easy to write Python interpreter code. So interpreter code, generally you just type a line and see what happened. But you can type in multiple lines, and the interpreter can handle that very smoothly. And if you find out that these few lines worked, just write them into a file and just load the file, and everything gets executed. So the compiler in the form of program files works quite well. And I say the Python string and list methods are very extensive and very easy to invoke. And I find one interesting thing is that usually if you do complicated things in Python, you would write new definitions, colon definitions, and all this thing to make nice subroutines. And I found that it was not necessary. So write small chunk of code in files, and then you can load many files from another file. And so this thing makes it almost unnecessary to define new functions, because most of the functions are methods, and you just invoke the method. And at least in my code, I didn't define any new definitions. I just make a file. And if I have to recall that all the function in a file, I just call it. Okay, so it's not necessary to define new functions. But that's just my preference. So here I just listed the code, the Python code that I've developed over the couple months. So the first routine is format. So we have genome files in what we call the FASTA format, and it is basically text file. You have one line of the name or some annotation attributes of RNA, and followed by multiple lines of ACGT nucleotide. And this, because they are lines with catch return line feed separating these various lines. And it's not very convenient to handle large amount of data in separate into different lines. So the format just take out, read all the lines in a text file, and it finds a header. It will store the name in a field, and then it keeps on reading the data and store all the data into the text field. And this text field can be thousands of characters long. And I think the Python doesn't mind if you really have lots of data in a string. And usually I use Excel to display this result, and Excel doesn't mind that either. So it's very nice to convert all the data into a file with two fields, name and nucleotide. And then in this format, I can manipulate the entire database with thousands and thousands of RNA in the data. And trim is mainly to remove redundancies. And I will talk more about redundancy. But the most obvious one is to read when you have everything in a big list. You can tell either Excel or Python to find all the duplicates and remove them. Okay, so trim at the very easiest portion is to remove all the duplicates. But there are redundancies which are not exactly duplicates. It's partial duplication, and that's the main problem I want to talk about this morning. And then once you have trimmed, you have cleaned up the data file, I would run a split. Split, it just saves the header into one file and save all the data into a data file. And in the header, I have the name and a pointer so that I know I have a list of all the RNA with their name and their position in the big data file. So the data file is a flat data file. It's just ACGT with no character return line feed, just a big flat file. But if you want to find out individual RNA, you have this index file, which has the pointer pointing to the RNA data stored in the data file. So it is very important to split the data, split it into data file and index file. So this big flat text file, I can do search, I can do all the manipulation very easily, because if you want to do it by individual RNA, you can do it, but it's much more involved in switching from different RNAs. So my basic strategy is to collect all data into a flat file, and then find all the patterns. And after I find the patterns, I would reinsert the index file to display the patterns in each individual RNA. So that's a program split. And repeats is the center of the analysis. It just go into the big, huge data, flat data file, and find all the repeats. Repeats are 20 nucleotide patterns, which are somewhere repeated in the entire data file. So I need to find all the repeats. And once you have found all the repeats, and I need to sort all the repeats according to their pointer in the data file. And once you sorted then you can find a lot of the repeated pattern are contiguous or consecutive. So many of the repeats are just next to each other. Their address has an address difference of 1. So if you have two patterns that's differed by one nucleotide, the second one is the continuation of the first one. So you just find out all the consecutive repeats and collect them into one big pattern. So this this contiguous string of nucleotide, I call them pairs. Pairs are variable length string, and this variable length string most likely are repeated somewhere in the data file, but not necessarily, because they are packed from neighboring repeats, and these repeats may not be the actual belong to an actual valid pair. So the pack. So I find lots of pairs, and from 20 nucleotide to thousands or 10,000s of nucleotide. So there are lots of these very long pairs, and the very long pairs, there are two possibilities. For one thing is that they could be a gene, okay? So they could be a gene which produce an RNA which is functional. Sometimes it might be used to produce a protein, but I believe there are probably lots of the RNA genes which are not used to produce protein, but they have their own functions. But the worst fear that I have is that if you have two RNA which are repeat, or two long stretches in two different RNA or in the same RNA which are repeats, and so if you have a N-nucleotide repeats, it will generate two N. You have two long stretches of repeated patterns. Then you have two N repeats, which are bogus. And if you include all these bogus repeats in your analysis, you basically have invalid pairs. So select is try to remove as many of these long, very long pairs. Try to find out if they are repeats themselves. And you have to remove the duplicates to make sure that the repeats are valid. So here in the big pair file, I would just try to find if the pairs are actually duplicates. If I find duplicates, I remove them. And so in the pair file, I try to identify valid pairs, and then these valid pairs are backtracked into the original data file and try to remove the RNA which causes this redundancy. So, and then if you go back, remove all the RNA which causes duplicates in the per file, and then resulting in a cleanup. So I will clean up the original data file, removing all the RNA which causes bogus repeats. So once I have removed all the bogus repeats, then I'll do the analysis again and try to see if I can clean up all the very long pairs and have more meaningful pairs resulted from the clean up the data. So the third time, just do exactly this. I'm sorry. This is not right. Okay. So the Python code number 3 is a repeat of the code 1. Okay. I just have a cleaned up data file to analyze. Sorry about this file. I haven't. So here is the biggest problem I have already mentioned. It is a duplicated long non-coding RNA produce huge amount of bogus repeats, which invalidates matching patterns. So if there are it is very difficult to identify and remove these nucleotide sequences. And let me see. So let me review the pattern search strategy. So the first, of course, you have you get your data into the proper shape, and then you find all the 20 nucleotides repeat patterns as repeats, and then you consolidate the adjacent or consecutive repeats to form a pairs. And then I believe that the clusters of pairs are very meaningful information in the RNA, and I call them necklaces. And hopefully, I think these are the programs these are the real code in the long non-coding RNA. So here are the 5 databases that I've used to analyze, and so the GRCH38. This is the Human Genome Project, okay, and they collect all the data. And you see the Human Genome Project. The main goal is to sequence all the chromosomes. But in this project, they found a lot of RNA, so they kind of collect all the RNA data in one piece, and that's called the GRCH38. And GRCH38, this is a human, okay, Homo sapien, and they collect all the non-coding RNA into one file, and it's 64 megabytes long. And another file which in the genome project is very interesting. It's called cDNA. So cDNA is mostly the RNA. They have they found some brain. I think these researchers use human brain, and they have they just take a brain, break it up, and extract all the RNA from the brain. And then they have there's an enzyme which can convert RNA into DNA. So the RNA is not very stable. So they get the fresh brain and extract the RNA and convert them to DNA, and the DNA is much more stable, and you can analyze the DNA at your leisure. Okay, so there are lots of data extracted from the human brain, and they are collected in the cDNA file. So, and I think because cDNA is produced from RNA, so I believe that there are lots of RNA. The cDNA is just RNA, so they contain the same information that I want to analyze. And beside this is a human genome project, but there are other people who are focused on RNA research. So there are other databases collected over the year. So Encyclopedia, this is a European group which catalog lots of different RNA molecules. And the non-code and also the link book. These two projects, I just searched the web and found them, and they have a very large collection of the RNA data. But then I read their website and found these two are all based in Beijing. And I kind of worried about these two databases because the other databases are collected by microbiologists. But these two databases are collected by computer scientists. So one group called the Big Data Center, okay, in Beijing. So I think they just work on their computers and collect data, and I don't think there are experienced microbiologists in handling all this huge database. And I think China, they just have big computers. They got lots of people, and they just do work everything on the computer. So the data they collected, I kind of don't have too much faith on them, but they have worked collect all these things. So it's something that I can work with. Okay, the redundancy in RNA. There are many. After I go through the analysis, so I got all the pairs. And the whole lots of them, and it is interesting that I found many very long pairs, and there are thousands or tens of thousands of nucleotide long, and these are as I said, if you have two long stretches, two long stretches which are duplicates. If you have two long stretches of nucleotide and nucleotide long, if they are duplicate in one or many different RNAs, and they will produce two N repeats, because every one, will be repeated. So in my analysis, I will collect them. But this may or may not be valid pairs. So I would all these repeats in these long stretches of duplicate nucleotide, and this I would call them bogus repeats. So they may or may not be valid pairs. So I think it is. I have to find a duplicate. If I have two of these long stretches, I have to eliminate one of them. Okay. Only after I can eliminate all the repeats, I can be sure of this long stretch of nucleotide or a valid pair or valid sequence that has a real meaning. So so last month I was mainly working on the redundancy removal. So the redundancy is that in the pairs or in the RNA, there are long stretches of nucleotide which are repeated in one or many RNAs. So it is, when you look through these files, it's easy to see them, but it is very, because the volume is too big, you just cannot go through them manually and identify them, identify repeat, and remove the repeats. So you have to do it automatically. You have to use computer to find all these and try to eliminate them. So it is quite very tedious, and it takes lots of computer time to find these repeats. So you see, it is very easy to find if two duplicates RNA. If they are duplicate, it is very easy to say, just tell Excel can do it. Excel has a checkbox that say remove duplicates. You just check it and it will remove all the duplicates very easily. But what happens if the repeats are not exactly the whole thing from end to end? If it's shifted by one nucleotide, you're in trouble. You cannot find it. Okay? If two big repeats, they are just displaced by one nucleotide, and then you cannot use the remove repeats in Excel or in the Python. You just can't find it. So, and I've, if everything begins at the same point, I say, okay, now look at the 200 of them. If two RNA has the same sequence for 200 nucleotides from the beginning, then I can find them very easily. If they are spaced by one, no, I'm out of luck. So finally, I found that the way to do it is that I have lots of long pairs. I want to find out if they are duplicates, and the duplicates is not at the same position. They can be displaced within the pair. So finally, I use the criteria that I go to all these long pairs, and I say I go to the middle of it and take 200 nucleotide, and then use this 200 nucleotide to compare with all the other long pairs to see if this 200 sequence appear in any of the other pairs. So the 200 nucleotide is a very long, But so if any two RNA has a stretch of 200 nucleotides which are identical, they might be in different positions, and I can locate them. Okay, so I go through the all the long non-coding and all the long, very long pairs, and generally I'll scan from the shortest pair to the longest pair. And in all the pairs, I would take the middle 200 nucleotide pattern and go through the rest of the pair list to find if there's anyone which has a match. If I find a match, then this one is repeat. So this one is eliminated from my pair list. So going through all the long pairs and find out if any of them has 200 nucleotide match, I would delete them, consider them to be repeats. So this way I can delete a lot of repeats, which I think could cause the bogus repeats. And once that I have this pair list cleaned up, then because I have all the address in the index file, so this pair list I can backtrack to the original RNA list and remove the RNA which contains these redundancy. Okay. So and once you remove all this redundancy, I can do the analysis again, and for sure the long non-coding RNA gets cleaned up. So I think this way I can get a list of pairs which are more meaningful in the final analysis. So the redundancy removal, I'm just showing the RNA, the number of RNA in the original data file. There are tens of thousands of them, and the pairs derived from them are in this column. And from this list of pairs, I would select things which are not repeated, not redundant. Okay, and you can see there's lots of being removed, and then these pairs are backtrack into the data file, and then you can see here the initially RNA we have 268,000, and once you remove the redundancy, there's only 81,000 remain, and then this number of pairs resulted from that. So I think this way the data, the pairs are much more meaningful. So here I'm going to show you 3 screenshots of the data. So one screenshot is the database as I formatted in the name and the sequence format, and I'll show you going through this file the first time. You see lots of pairs, but lots of pairs are very long ones, which I think are redundant. And then I'll show you the a file which, after I remove the redundant and the per list, looks much more, much cleaner. And I hope that these things are. Yeah, this is the eyesight test. So this is the original data. This is the first column, which are the names that used in the database. And these are just the data. And this data goes on and goes. You have tens of thousands of bases. But Excel is very nice. It just, if there's nothing, if there's no other columns with data, it just displays the last column to the end. So it's very, very nice to look at. At least you can see the first hundreds of the nucleotide. So in this database, the first run around, this is the per list. Okay, so the first column is the address in the big database. These are all the pers, and this is these are the indexes taken from the index file. And you can see these pers, and this is the length of the per. And this is the length of the RNA molecule. So here you can see this guy is 2,787 nucleotide long. So that's a very long nucleotide and a very long per. And I believe this contains a lot of bogus repeats. So it can actually have 2767 bogus repeats contained in that long per. So these are the target that I want to say. I want to look up in the data file. So where is the other per which causes this one to appear? So I want to go through the entire thing and find out which other one causes this repeat. And if I can remove all the other ones, this one would give me valid information in terms of the pers. Okay. So there are short ones, there are long ones, there are very long ones, and this I think may be useful or may not be useful, but only after I removed all the redundant, or what I think, or what the computer can see, all the pers which has repeated 200 nucleotide. So once these repeats are removed, you can see the per list cleaned up quite a bit. Okay, so they are short lists, and these are what I feel are code in the long non-coding RNA. And so, after doing all these things, I'll say, oh well, I have to look at this RNA database more critically. I got 5 huge collections, and I need to be sure that I have something that actually meaningful, actually useful. So I did an intersection analysis to look at all these data files and see what they are the same and where they are different. So the intersection is just take two big database and look at the matches between the two databases. So here, these are the diagonal column is just the RNA molecules in each database, and you can see between these two databases there are 14,000 prove this grand scheme of cell computer. So the microRNA, I have a long time ago, I've downloaded microRNA, and this microRNA actually contains microRNA from 27 different species. Okay, so it it cataloged 48,000 microRNAs, and I went through the RNA list and do the same kind of inspection and found that only 27,000 are unique. Okay, and I would use, and among these 48,000, only 5,312 are from human. So this is a very limited collection, but I would like to see if there's any microRNA in the human long non-coding RNA databases. So I went through all these big databases, and these are all the long non-coding RNAs, and I would have this 27,000 microRNA database and to see if there's any match. Yeah, there are match, but not very many. So the matching record is that if any occurrence of the microRNA in any of the RNA, so for example, in this huge database, I found 10,000 matches. And among the 10,000 matches, the microRNA, there are 532 microRNAs has matches in this database. And then there are 8,000 long non-coding RNA which contains one of the microRNAs. So there are matches, but not enough. So I can say I have proven that microRNA are the actual I cannot claim that I prove this microRNA are in the linear long non-coding RNA, and thus causes all the subsequent mechanism to work. So that's as far as I get. I would like to see that there are long non-coding RNAs, and long non-coding RNAs has lots of pairs, and many of the pairs are actually microRNAs. That's what I want to see. But I think as you can see, the database in the human long non-coding RNA databases, they are still inconsistent, and I don't think they are complete. And microRNA database is very limited. So we only knew a very small number of RNA, and so with two very incomplete databases, of course I cannot prove anything. I would just hope that the more we know about microRNA, and the more we know about the long non-coding RNA, or somehow in the future we can make the connection. Okay, but now it's just a proposal. It's a crazy idea, and I just hope that I would do my best to analyze these long non-coding RNA data files. And I think this analysis will be very useful for microbiologists to look at the database to see if they have something that's it can validate their data. But that's the only useful thing I can see my analysis can contribute. But I would hope that in the future, when we have more RNA data and more microRNA data, we can make a better connection. Okay, that's all I have. Thank you very much. Okay, any more questions, or we can go to lunch?
About these database.
Yes.
Could you ask him at lunch? Because
Right, well, okay.
it's homecoming and we'd like to get there.
Oh, yeah. Okay,
Do that because
okay.
it has to do, it's just the RNA is what it
Yeah.
is, right?
All right. Okay, thank you very much. See you after lunch.
Can you guys bring us back something you want to